many thanks for the invitation to come speak about my work, which is on the topic of noise in the nervous system. And so what I'll do is I'll start by illustrating what I mean by noise, and then we'll see sort of what the core ideas are behind my research. So the example here is a visual one. You have two eyes that gather information about the world, send that information down the optic nerve to your brain. And to take a closer look at that, we'll pick out one fiber of the optic nerve, that is to say the axonal output from one retinal ganglion cell. And in particular this cell, which is a special type called a direction-selected retinal ganglion cell, for reasons you'll see in a moment. And it's the orange thing in the middle of this picture. We'll consider an experiment where my collaborator Max attaches tiny wires to the membrane of this cell and then records the membrane potential as a function of time. Right? And you see that the membrane potential is largely near this baseline value of negative 65 millivolts, but occasionally that quiescence is punctuated by these brief excursions that we very imaginatively call spikes. Right? And these spikes travel down the optic nerve and they carry the information to the animal's brain. Yeah. So these are loose patch cell-attached recordings. So he attaches a pipette to the cell membrane, sucks in a little bit of the cell membrane without breaking it, and then has a tiny wire inside that pipette.
So it's almost like a patch clamp but with a wire.
It's a loose patch. Okay. So now to get at the idea of noise, we'll consider the following slightly more complicated experiment where Max is flashing a movie onto the retina. The movie is shown here schematically, and it consists of a bar of light moving in different directions. So left, right, up, down, and so on. And while the movie's playing, Max records the spiking activity of the cell, which is shown in this raster plot below. In this plot, each of these little vertical bars represents one spike, and each row is the response recorded from the cell on one repeat of the same stimulus. So we'll look in one particular window, so say the period of time when this downward moving stimulus is present on the retina, and then make a histogram of the number of spikes emitted by the cell each time that bar passed over the retina. And this is what you see, and there's an enormous amount of variability. Sometimes the cell emits around 20 spikes, other times close to 10, and other times approaching 30. Huge range of responses, all associated with the same stimulus. This is not a strange quirk of the retina, nor of the direction selective system we're looking at. You see similar types of variability, Poisson-like variability, variance approximately equal to the mean, in places like cortex, hippocampus, auditory system, and so on. But what this means is that because the brain's information about the scene comes from the spikes traveling down the optic nerve, and those spikes are not very reliably locked to the stimulus, it means the brain is getting noisy information about the environment. For contrast, we have a very robust ability to perceive the environment. There's a classic experiment that dates back over 70 years now that's sort of shown in a loosely described form by this picture, where Hecht had human subjects sit down in a chair, look at a monitor, and then he asked them to report by pushing a button when they saw a flash. And what he did is turn down the intensity of the flash and saw what the minimum flash intensity was before subjects failed to report it, and found that the dimmest flash he could show that would still be reliably reported was so faint that it elicited only between 1 and 10 photoisomerizations of the photoreceptors. So you can detect such faint stimuli that only 1 to 10 photoreceptors are triggered, despite having this enormously high level of noise in each of the channels sending information down the optic nerve. So in a very big picture sense, what my research program is about is trying to identify the source of the nervous system's emergent robustness. How do you take a system of unreliable components and construct something that is robust? Okay. So what I'll do today is outline the specific way we approach this. I'll start with some theoretical ideas about the structure of signal and noise, and I'll eventually apply those back to our recordings from the mouse retina. So, and I realized at lunch talking to Tom Schneider that I maybe use the word noise in a way that's a little bit atypical from the perspective of information theorists. So what I mean is that the message, so the spike train traveling down the optic nerve, consists of some signal, which we'll define to be the mean message averaged over repeats of the same stimulus, corrupted with some zero mean noise. And this is what I mean by noise. It's everything that is trial-to-trial fluctuations about the mean signal that the cell is trying to transmit. And we'll guess then that the ability to robustly transmit the signal will depend on the relationship between the signal and the noise, and we'll tease that apart. See what I mean by signal structure? We'll imagine a simple example where the eyes are sending a picture to the brain, and those pictures come from some sort of artificial world that I just made out here. Each of these little blocks represents one picture drawn from the world, and so this 8x8 grid has 64 of these sort of hypothetical pictures. And what you see by looking at them is these aren't kind of random white noise type images, but they instead consist of white bars, either vertical or horizontal, all superimposed on a black background. Now, if I give you a noise-corrupted version of one of these images, so imagine this being the message received by the brain, you'll notice that at least in this example, you can easily guess what the most likely true message is, the most likely underlying signal. It's the following. And the reason you're able to do that is because the noise that I've corrupted the image with doesn't consist of these white extended bars on a black background. It's uncorrelated Gaussian white noise, so it's these very small pixel by pixel fluctuations, and that has a different statistical structure from the signal, which allows it to be separable, similar to the idea of multiplexing. So to understand this communication problem between retina and the brain, we need to quantify the structure of the noise in the retinal ganglion cell responses. So that's the noise in the actual messages going to the brain. We need to quantify the signals, and then we need to look for something like this, wherein there's a different structure to the signal versus the noise. What is the noise structure? Well, we'll consider now the following experiment, again done by Max. Here Max is doing these loose patch recordings simultaneously from two of these direction-selected retinal ganglion cells, again while he projects this movie onto the retina. And so what you see here is the raster plot for each of the two cells over repeat presentations of the same movie.
Is the movie done in extremely low light conditions?
This is done relatively dark, yeah.
So
And
what's
this is,
the relevance of the first example where you had the single photon detection? You can't detect single photons if you're sitting in a room like this.
That's right. So these are dark adapted animals. So in particular, these are done on disembodied retina. The animal spends the last day of its life in a dark box with no light. It's sacrificed in the dark. We actually wear night vision goggles in the lab. There's no ambient light. Remove the retina, place it under a movie. The movie is also quite dim. And you have to do this so you don't just bleach all of the photosensitive pigments on the retina, rendering it blind. Otherwise, you can't do this sort of experiment. So this is relatively low light. Although again, if you look in cortex, even in quite bright light conditions, you also see these high levels of variability. So I think the problem is quite general, and we're approaching it in the sort of way we have the technology to do it. Okay. So we're quantifying the noise structure, and so what's shown here is the response of the two cells, number of spikes emitted by cell 1, number of spikes by cell 2, on repeat presentations of the stem. And each dot here represents the response on one trial. And you'll notice first the noise. The little dots are far from the center of this cloud, so there's variability. And moreover, the variability is extremely tightly correlated between the cells. On trials where cell 1 gives too many spikes, cell 2 also gives too many spikes, typically, and so on. So these are called noise correlations, which is just the observation that these trial-by-trial fluctuations are correlated between cells. And these also, much like the noise itself, appear to be quite ubiquitous in the nervous system. If one looks in retina, in the thalamus, in visual cortex, in inferotemporal cortex, and so on, we typically observe non-zero noise correlations. The sort of restating of the question we started with then is how this collective phenomenon of correlated variability affects the robustness of information transmission, in particular by the retina. And as I've stated, and we sort of illustrated with this bars example, this probably has something to do with the signal versus noise structure. We sort of know the noise structure, it's correlated noise. The question about the signal we'll get by taking the responses to each stimulus and just averaging over trials to get that mean response that I've defined earlier as the signal. For the two cells shown here, these are the mean responses. These are called tuning curves. They tell you the average number of spikes emitted by the cell over any presentation of a particular stimulus. And in this case, the direction selectivity of the cells is quite obvious. The mean response is strongly modulated by the direction of motion of the stimulus. So for our communication problem, we have these means, these signals, that then get corrupted by a noise that's correlated between cells, and that then gets received by the brain. So what we'd like to understand, and we'll start with a theoretical problem, is how the correlations in the variability itself affect the brain's ability to estimate the stimulus after receiving these responses from the retina. To construct our theory, we will allow for arbitrary tuning curves, just shown a bunch of sort of random, vaguely Gaussian-ish shapes on this plot, but it doesn't matter. We will assume arbitrary noise variance for each cell, which again won't turn out to matter. And then what we'll do is we will try to compute how correlations affect information transmission for fixed arbitrary tuning curves and fixed arbitrary noise variance. Right. So we'll be a little bit more mathematically precise now. We'll define our response vector over the population of cells as a vector of the mean responses, f of s. F of s then is the tuning curves, right, that give the mean response to any stim, s, then corrupted by some vector of noise. We'll then ask the estimation problem, what after receiving the vector of responses r, how well can we estimate the stimulus s? In other words, how well can the brain recover the direction of motion? And we'll quantify this with the linear Fisher information, although I'll mention that the results in this paper of ours now in PLOS Computational Biology also show similar, well, they show exactly the same trends for mutual information, as long as you make some assumptions about Gaussianity, which I know offends Stefan, but this is what we did. So what is this Fisher information? It's the following quantity. It depends on derivatives of these tuning curves with respect to the stimulus S, and then the inverse of the covariance matrix of the responses conditioned on the stimulus. And geometrically, it has the following sort of flavor. We imagine that we showed some stimulus, stim1, and got some set of responses defined by this green ellipsoid. We then present some other stimulus, stim2, and get some other ellipsoid of responses. And we'll imagine the two stimuli are similar, so it's a small change in the stimulus. What the Fisher information tells us is how sensitive the responses are, so how much the centers of these clouds, or the mean responses, change relative to the amount of variability in the responses along this direction. And you can see this formally by diagonalizing the covariance matrix, considering projections of these things onto the eigenvalues, or eigenvectors rather, the covariance matrix, and so on. And so now to construct our theory about correlations, we'll imagine that there are some tuning curves that are fixed, some diagonal matrix in the covariance that is also fixed, that contains the variances in each cell's response, and then some off-diagonal matrix B, a hollow matrix, zeros on the diagonals, multiplied by some parameter t, some scalar parameter. We'll take derivatives of information with respect to t, in other words, take directional derivatives in the space of matrices of information with respect to those off-diagonals, and then ask what structures of this matrix B make that derivative positive. In other words, what types of off-diagonal elements, what types of correlation coefficients make for increasing information about the stimulus in the neural responses. So there are a lot of specific results in that paper that are quite technical. The general picture, though, I think is quite intuitive, and it's the following. We'll consider the abstract space of neural responses, and consider the set of responses to some particular stimulus, shown here in blue. And then we'll consider the responses to another stimulus, and another stimulus, another one, and another one. And what you see here is the response clouds are displaced from each other, so their means are displaced along the skinny direction of those clouds. And if you think about Fisher information, that means that your sort of change in response divided by the variance in that direction is relatively high, because there is relatively low amount of noise along the direction by which the center of the clouds are displaced. For contrast, if we consider a different situation where the response clouds are displaced along their long axes, this corresponds to a case with relatively low Fisher information, because there's a lot of variability along the axis of change of the centers of the clouds. So the way we sort of think about this then is we draw this green curve, which goes through the centers of all the clouds, So the signal curve defines the set of mean responses over the population to any stim. And the geometrical thing we're looking for then is that if the signal is orthogonal to the noise clouds, so if most of the variability is orthogonal to this curve, that's good for information propagation. If, for contrast, the noise clouds are largely parallel to that signal space, that's bad for information propagation. Sorry. All right, so to make progress then in connecting these ideas to neurobiology, we'll need to measure this green curve, the signal space, and then look for how the noise is oriented relative to that in the space of neural responses. This is unfortunately quite hard. If you think about trying to measure this curve, what you need to do is show each stimulus many, many times to get the average, and then show many different stimuli so that you can construct this whole set of points. In practice, you can only record from these cells for a few hours before they die, and so we can't just by brute force sample this signal space. But we get lucky by considering these special cells that we started with, which are the direction-selective retinal ganglion cells, for reasons that I'll describe in a moment. So recall this is the tuning curve, so mean response to any stimulus, and that's the data are shown in black on this plot. Overlain on that is this sinusoidal function. So the mean response, and this should really be an F, I apologize, is some amplitude factor A times some constant offset B plus a sinusoidal function of the stimulus angle. The phase phi tells you where the peak of this tuning curve is, and that's what's called the preferred direction of the cell. So the stimulus direction for which you get the most spikes. And the amazing thing about the direction-selective cells, and this is a discovery that goes back to Barlow in the 60s, is that they come in 4 subtypes. So each subtype has a preferred direction in one of the 4 cardinal directions. So you have up-selective, down-selective, left and right-selective DS cells, but none that are northwest selective, for example. Now, if we do a little bit of trigonometry and we make the following assumptions, our life simplifies greatly. So we'll assume we have the same number of cells in our neural population, so the set of cells communicating with the brain. So say 1 each of up, down, left, right. We'll also assume that the tuning curves are symmetrical. So they'll have the same shapes, but just be shifted each by 90 degrees along the stimulus direction axis. If you do that, then you can use simple trig identities to note that the mean population response to any stimulus direction theta will always lie on the surface of a hyperspherical shell. This is like sine squared plus cosine squared equals 1 type of trig. What this looks like then, and I'll illustrate this with an example of 8 cells, 2 each of up, down, and left, right, slightly displace the tuning curves for each of the identically tuned cells just so you can see them, is the following. So we'll present some stimulus, so some direction shown here by our blue bar. The mean response of the population will then be given along each of the coordinates in the 8-dimensional response space by the intersection of this blue bar with the appropriate tuning curve. We'll then consider a different direction of stimulation shown in red, and you then get some other mean response over the population shown by this red arrow here. And the geometrical fact is that both the blue and the red arrows have their tips on the surface of this hypersphere. So from our theoretical perspective then, we get the following picture about what sorts of noise configurations will or will not contaminate the direction message being sent to the brain. In this example, which is hard to see, the variability is largely radial in the space of neural responses. So if you present the same stim over and over again, you might get this response or this one or this one. And that means that a recipient can normalize the response vector that it gets, so project it onto a unit hypersphere, and as a result, obviate most of the variability in the neural responses. So that's this blue configuration where noise is in the safe direction that's not problematic for the neural code. For contrast, this sort of purplish setup is one in which the noise is spread out tangentially along the surface of the hypersphere. And in that case, the noise does strongly contaminate the information transmission. So we'll guess on theoretical grounds that we should see noise that's largely in this radial direction if the retina is sort of set up so as to effectively communicate with the animal's brain. These are what our sort of data look like. Again, this is for 2 cells. Each of the different colors here corresponds to a different direction of motion of the stimulus. The stick in each case goes out to the mean response, averaged over trials, These ellipsoids are the one standard deviation contours of the conditional response distributions. What we're looking for in asking about radial variability is for the long axes of these ellipsoids to be roughly parallel to the sticks that they're attached to. And this is what we see for this particular cell pair. To summarize our data over all of our recordings, not just this one example cell pair, we'll quantify the tangential variance, so variability along this direction that's orthogonal to the stick. We'll then estimate the radial variance, amount of variability in the direction that's parallel to the stick, and construct the following metric, which is radial variance divided by radial plus tangential. So if this number is 1, all of the variability is in the safe radial direction. If the number is 0, all of the variability is in the dangerous tangential direction. The vertical axis here are the raw experimental data. The average value is roughly 0.8, so 80% of the variability in the safe radial direction. Now, to get back to the question of correlations, what we've done is constructed on the horizontal axis here uncorrelated surrogate data. So we've done this by trial shuffling. So take all the responses of the two cells to one particular stimulus, say the down-moving stimulus, and then take trial 1 of cell 1 and trial 2 of cell 2, stitch those together and pretend that they were simultaneously recorded. And then repeat that shuffle over all possible permutations. And so what you get then are surrogate responses, an uncorrelated model for which the marginal statistics are exactly preserved for each cell, but the correlations are set to 0. And so for the uncorrelated model data, the fraction of variance in this radial direction is significantly lower than it is for the true measured experimental data. This is to say that the correlations in the variability orient noise in this safe radial direction where it's out of the way of the information transmission. Done some analytics to show that these observations in pairs of cells suffice to show that if we had looked at a full set of, say, 8 or 16 or something of these cells, you would also have variability largely radially oriented in that n-dimensional response space. Okay, so we've built a geometrical picture about orientation of signal versus noise that is or is not good for population coding. We've seen that our direction-selective cells in mouse retina appear to orient noise in the safe direction, and what remains to be done is to show empirically that by actually decoding the responses to estimate the direction, that there actually is the effect that we think is there. But we immediately run into a problem here, and that's that our experiments are done by recording from pairs of cells, so 2 at a time. For contrast, we know for anatomical reasons that we need to consider populations of at least 8 cells at a time. In particular, if you consider the number of retinal ganglion cells that each convey direction information about one particular spot on the retina, that number turns out to be 8, 2 each, two up, 2 down, 2 left, 2 right, for each little spot on the mouse retina. So we need to consider populations of 8 of these cells. We've only got recordings from 2, and we need to consider the joint statistics, so at least pairwise correlations, and correlations of pairs at a time don't suffice to construct the 8x8 covariance matrix. So what we did is a lot of intracellular recordings that I won't talk about that allowed us to build a model of the actual circuitry underlying these responses. And then I can take 8-cell models and do whatever I want with them once I've built those. The model has a small number of free parameters that I fit using a genetic algorithm, and the goal of the fit was to match the distributions of response statistics that we actually recorded in pairs of cells. So make a model of 8 cells, tune it such that if you pick out any pair of cells from the model, the distributions of statistics on those pairs match the recorded pairwise distributions. We'll now consider some held out data that was not used in training the model. That's what you've already seen, radial variance fraction for the correlated versus shuffled responses. The black is the experimental data and the blue is the model. And what we're looking for is for the distributions of these dots to overlap. And you can do things like a KS test, 2-dimensional Kolmogorov type test, and see that they're not different from each other. Okay. So we have a way to look at responses of 8-cell populations that are consistent with all our pairwise recordings. What we'll do now is compute the information in these model 8-cell population responses about the direction of motion of the stimulus. we get the following. So we've done this with two different estimators, a nonlinear maximum likelihood one and then a linear estimator to check that our conclusions are robust. The vertical axis here is your precision with which you can estimate the stimulus, so high values are good. The red is the precision obtained with the correlated responses. The yellow is the precision obtained with uncorrelated trial shuffled responses. And so the factor of 2 difference between the red and the yellow bars is the statement that the correlations in these responses, that collective phenomenon, which we've seen orients the variability radially in the response space, roughly doubles the ability of these 8-cell populations to encode direction of motion of the stimulus. Now, this is perhaps a little bit unsatisfying because we've done it indirectly by constructing this 8-cell model. I'll mention that recently some colleagues of ours in Europe have used array methods to simultaneously record from 8-cell populations of these DS ganglion cells and see like exactly the same picture that we've constructed. So this is not sort of artifactual of the way we've constructed our model. So the summary, we started with this question of how do you take noisy single-cell responses and construct a system that has emergent robustness. We started by theorizing that correlations, so collective activity, will improve the system's robustness if they orient noise in a direction that's orthogonal to the signals the system is trying to transmit. We then considered experiments from the mouse retina where we showed that correlations orient noise in this radial direction, which is orthogonal to the hyperspherical shell along which the signals actually lie. Work that is in progress or almost done now, I'm doing on asking what kinds of circuit mechanisms, so patterns of interconnectivity between cells, can actually shape noise in these beneficial ways so that you get emergent robustness in the population codes. We've also only done this for one specific type of retinal ganglion cell, that's these direction selective cells. There are about 20 different types, and so we're working hard on applying these ideas to things like the on-alpha ganglion cells, and so on. So many thanks to the people that contributed to this work. The recordings in retina were done by Fred, Max, and John in our medical school. The theory about correlations and coding I constructed along with our students, Yuhu, Alex Kaiko Gaich, and my mentor, Eric Shea Brown. The crew at Case Western contributed hippocampus recordings that I unfortunately didn't have a chance to talk about today. But if you're interested in reading more, you can check out my website. Thanks. Yeah.
The question is, how would the system orient the noise? What I'm thinking is that maybe you could turn that around and say the noise is in some direction and the system evolves to match that direction.
Yeah, so this is exactly the way I'm thinking about generalizing this observation to other cell types. So with DS cells, these direction selective cells, we got lucky that we could use geometry to find this spherical shell. But for other subtypes of ganglion cells, there's not such a clear sort of signal space. But what I'm trying to do is to figure out, if we record responses to one stimulus and we get clouds that look like this, and then another stimulus, we get clouds that look like this, well, we can guess that the set of things that the population can actually transmit will lie along the curve that's orthogonal to the long axes. So we can almost use the noise properties to guess at what the signal should be. And this is work that's in progress. Yeah.
So what would happen if there was no signal, nothing flashed at all? Like it was just pure darkness. Is there a measure of noise there?
So these cells turn out to be remarkably, I'm not sure quite what the word is, but if you shut off the stimulus, they really are silent. There's not like a lot of sort of dark current or spurious spikes in the absence of a stimulus.
Poppy is waiting for us, so